Hello students, welcome to EPG Pathsala. I am Ramesh Kothari, Professor of Microbiology, UGC CS Department of Biosciences, Saurashtra University, Rajkot, Gujarat. Welcome you all. Today we are going to discuss about gluconeogenesis from the paper Metabolism of Carbohydrate. The synthesis of new glucose, which is known as gluconeogenesis. Gluconeogenesis is the synthesis of carbohydrate from non-carbohydrate precursors. These precursors include lactate, that is pyruvate, and some amino acids called glucogenic amino acids. A small amount of glucose can be made from odd chain fatty acids too. For the most part, gluconeogenesis is confined to the liver. Under normal circumstances, the liver is responsible for 85 to 95% of glucose that is made. In starvation or during metabolic acidosis, the kidney is capable of making glucose and then may contribute up to 50% of the glucose formed. Since in these conditions, the amount of contributed by the liver is decreased considerably. The only other tissue capable of gluconeogenesis is epithelial cell of the small intestine. The significance of this activity is not clear and it contributes at best not more than 5% of the total glucose formation. Now let's see the objective of this lecture. In this lecture, we will discuss how glucose is synthesized in our body via non-carbohydrate intermediates. We will also study the fate of various carbohydrate substrates in our body and overall glucose requirement by various cell and tissues. Thereafter, we shall see how energy efficient the reactions of glycolysis and gluconeogenesis are. Further, we will also see a detailed gluconeogenesis, that is generation of new glucose in body and how the normal glycolysis pathway is bypassed when body is under starvation or long duration of fasting. We will also discuss the various entry points, precursor molecules, and the fate of various intermediate, and at last but not the least, we shall see the actual biochemistry behind these reactions and role of various biocatalysts in gluconeogenesis pathway. The figure shows the concept map of gluconeogenesis. As you see in the concept map, there are various junctions from where precursors allow to enter the central metabolism. Amino acid intermediates, glycerol, and fatty acids are able to ultimately transformed into glucose via gluconeogenesis. If you see the entry point, Calvin benzene cycle, glyceraldehyde triphosphate. From amino acid from protein, there are two entry points, that is at glyceraldehyde triphosphate and oxaloacetic acid. From glycerol, that is from fat, there is again dihydroxy acetone phosphate. Then from a fatty acid, again from fat, that is to acetyl-CoA. Now this acetyl-CoA is during this gluconeogenesis converting to pyruvic acid to oxaloacetic acid, glyceraldehyde triphosphate to fructose 1,6-bisphosphate to fructose 6-phosphate to glucose 6-phosphate to glucose, and ultimately in the starch and cellulose. Again here the glucose 6-phosphate act as a precursor for glycogen synthesis, and likewise the fructose 6-phosphate act as a precursor for the peptidoglycan. Gluconeogenesis is an ubiquitous process observed in all of the living kingdom, including plant, animal, fungi, and bacteria. This process is also referred to as an endogenous glucose production. Gluconeogenesis is responsible for glucose generation from non-carbohydrate substrate, as mentioned earlier. The preliminary carbon skeleton in gluconeogenesis is mainly from pyruvate, lactate, glycerol, and the amino acids alanine and glutamine. This figure is a schematic representation of various pathways of carbohydrate metabolism, metabolic junctions, precursors, and storage reserve among plant and animal. If you see in this schematic representation, various intermediates enters into the central pathways through various routes, irrespective of plant or animals, for storing energy reserve or for generating energy from reserved substrate. Discrete pathways are designed. Various stored reserves are glycogen, starch, glycoproteins, disaccharides such as sucrose, and monosaccharides such as blood glucose. If you see in the figure that in animal, lactate is converted to pyruvate, and then it is getting its route through the TCA cycle. Likewise, from plant, the carbon dioxide fixation results in the formation of 3-phosphoglycerate, which again enters in the central metabolic pathway, and then it all results in the formation of glucose 6-phosphate, which ultimately becomes the node for the synthesis of blood glucose, glycogen, glycoproteins, disaccharides, or the monosaccharide, starch, sucrose, etc. Gluconeogenesis and glycogenolysis are the two mechanisms that help in maintaining blood glucose level in the body. Oxaloacetate and dihydroxyacetone phosphate are the key intermediate for gluconeogenesis as well as all other metabolic pathways. Cell, whether in normal fed state or under starvation, channelize certain reactions for glucose generation, and these are routed through pyruvate, lactate, oxaloacetate, acetyl coenzymes at one point or other. Two important reactions which help the cell to acquire amino acid intermediates, that is alanine and glutamine, and lactate from muscles, and can readily convert into pyruvate, glycerol or dihydroxyacetone phosphate are alanine cycle and corecycle. The reactions that constitute the corecycle varies, wherein a pyruvate is synthesized from lactate in muscle tissue, and in another reaction of transformation in muscle, alanine is formed from pyruvate. The amino acid group release is reduced in the form of urea. The reaction are popularly called alanine cycle. Both these corecycle and alanine cycle reactions allow generation of pyruvate and thereby favoring entry into gluconeogenesis. The hydrolysis of triacyl glycerols in adipocytes yield fatty acids and glycerol. Glycerol act as a precursor of glucose, but animals are unable to transform fatty acid residue to glucose. This figure shows glucose metabolism and regeneration reactions in liver and skeletal muscles, depicting corecycle and alanine cycle. The diagram A shows the corecycle, where transformation of lactic acid produced in skeletal muscles into glucose in liver cell via pyruvate intermediate, while in diagram B, amino acid intermediates formed by protein metabolism are transported to liver, where in a simple deamination reaction help to form pyruvate and glucose upon further hydrolysis. Deamination also produces urea as metabolic waste. There are three reactions which are thermodynamically not favored in reverse direction. These are reaction first, conversion of glucose into glucose 6-phosphate, which is catalyzed by an enzyme hexokinase. Conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, catalyzed by enzyme phosphofructokinase. And the third reaction is formation of pyruvate from phosphoenol pyruvate, catalyzed by an enzyme pyruvate kinase. Essentially, these reactions doesn't allow gluconeogenesis to proceed easily as glycolysis. The process of gluconeogenesis is not very cost effective from energy point of view, as oxidation of glucose through 2 mole pyruvate finally yield a mere 2 mole of ATP, while generation of glucose via gluconeogenesis consume at least 6 mole of ATP at various stages. The net deficit in terms of ATP in gluconeogenesis is equivalent to 4 ATP, as 2 ATP are produced and a total of 6 ATP are used up in generating glucose from pyruvate. The main reaction in gluconeogenesis is a conversion of oxaloacetate into intermediate in order to enter the pathway. The reaction is catalyzed by phosphoenol pyruvate carboxykinase enzyme. The reaction consumes energy in the form of GTP, that is energy equivalent to ATP. In the last reaction, wherein 1 mole of glyceraldehyde 3-phosphate needs to isomerize into dihydroxyacetone phosphate, and then further upon a condensation reaction giving 1 mole of fructose 1,6-bisphosphate, a reaction exactly reverse of aldolase enzyme action. This reaction of gluconeogenesis lead to generation of glucose in liver cell that can readily be added into blood circulatory system and act as a source of nourishment for vital body tissue. So, let me see the function of gluconeogenesis. During starvation or during period of limited supply of carbohydrate, when the level of liver glycogen are low, gluconeogenesis is important in maintaining adequate blood sugar concentration. During severe exercise, when high catecholamine level have mobilized carbohydrate and lipid reserves, the gluconeogenic pathway allow the utilization of lactate from glycolysis and of glycerol from fat breakdown. During metabolic acidosis, gluconeogenesis in the kidney allow the excretion of an increased number of protons. Gluconeogenesis also allow the use of dietary protein in carbohydrate pathways after disposal of the amino acid nitrogen as urea. There are 3 bypass reactions, and these reactions are the bottleneck reactions. Bypass reaction 1 is the conversion of pyruvate into phosphoenol pyruvate via oxaloacetate. Bypass reaction 2 is the conversion of fructose 1,6-bisphosphate into fructose 6-phosphate. And bypass reaction 3 is the generation of free glucose from glucose 6-phosphate. Bypass reaction 1. This reaction utilizes the energy from adenosine triphosphate. Enzyme pyruvate kinase requires biotin as a cofactor in presence of carbon dioxide. The carbon dioxide utilizes in this reaction occurs as a bicarbonate ion. This is the first reaction of gluconeogenesis process, and as the name of the enzyme suggests, the substrate pyruvate is carboxylated to form oxaloacetate. Pyruvate carboxylase is stringent in its requirement of activator. In absence of activator, that is acetyl coenzyme A, the enzyme becomes inactive. The chief source of acetyl coenzyme is beta oxidation of fatty acids in liver and adipocyte. The enzyme mainly function to generate carbon skeleton from non-carbon intermediate. Pyruvate carboxylase work for formation of pyruvate, lactic and alanine. Additionally, it works to derive oxaloacetate and thus tricarboxylic acid cycle. As shown in figure, the reaction occurs in two simple stages, one in which biotin is carboxylated to carboxybiotin in presence of bicarbonate and spending energy from ATP. Then through carboxyphosphate intermediate, it transfer the carbon dioxide to pyruvate forming oxaloacetate and regenerating biotin. The carboxybiotin is activated form and has standard free energy for its cleavage, which is equivalent to 4.7 kilocalorie per mole. This negative standard free energy indicates that carboxybiotin is able to transfer its carbon dioxide without any further energy input. This reaction shows a formation of phosphoenol pyruvate from oxaloacetate, a reaction catalyzed by phosphoenol pyruvate carboxykinase utilizing energy from GTP. There is no net fixation of carbon dioxide, as at the end of this reaction the carbon dioxide that was initially incorporated by pyruvate carboxylase into pyruvate is subsequently released by phosphoenol pyruvate carboxykinase. For gluconeogenesis to proceed further, the oxaloacetate must be transported to a cytoplasm, for which no mechanism exists in the cell. At the same time, the free diffusion is not possible. Three distinct reactions that help in this matter, they are bypass reaction 1, transamination to aspartate and reduction to malate. If you see in the figure, in liver cell, the alanine are converted into pyruvate and pyruvate is converted into glucose. At the same time, this deamination of alanine results in the formation of urea. Now the glucose which is synthesized in liver, it enters in the muscles where it converted into pyruvate, releasing 2 ATP, and from pyruvate it converted into lactate. From pyruvate it is also converted into alanine, which is an reversible reaction where alpha keto acid is converted into alpha amino acid, and alpha amino acid can be converted into alpha keto acid. And this alanine again enters in the liver through blood. So this pyruvate, when enters in the in the muscle cell, the pyruvate convert into oxaloacetate. When pyruvate is enter in the TCA cycle, it converted into oxaloacetate and malate. Then ultimately malate is converted into oxaloacetate to phosphoenol pyruvate to fructose 1,6-bisphosphate to fructose 6-phosphate to glucose 6-phosphate, and ultimately it results in the formation of glucose. This is a bypass reaction 2. It is a simple hydrolysis reaction and is catalyzed by enzyme fructose 1,6-bisphosphate, which convert fructose 1,6-bisphosphate to fructose 6-phosphate, releasing inorganic phosphate. This slide shows the bypass reaction 3. it is again a simple hydrolysis reaction and is catalyzed by enzyme glucose 6 phosphatase. Excess glucose should not enter the bloodstream as it is detrimental. So majority of glucose 6 phosphate is withheld in endoplasmic reticulum lumen and converted into glucose upon requirement. If you see in the figure, it shows the generation of free glucose from glucose 6 phosphate. In endoplasmic reticulum lumen, the when glucose is glucose 6 phosphate when enters from cytosolic side into the endoplasmic reticulum lumen, once the glucose 6 phosphate is inside the endoplasmic reticulum lumen, it converted into glucose releasing pyrophosphate. Now this glucose again can pass through this membrane and enters in the cytosolic reactions, cytosolic site. So hence the generation of free glucose from glucose 6 phosphate can be achieved by the enzyme glucose 6 phosphatase, which is present in the membrane of endoplasmic reticulum. So, dear friends, in conclusion, we can say that gluconeogenesis is less energy efficient, but at the same time, it is a vital process by which glucose synthesis is achieved. We know that there are many ways through which cell gets glucose, but this is one of the way through which cell fulfill its requirement of glucose. Key processes for de novo glucose biosynthesis and a lifeline for vital tissues and brain under longer duration of fasting and heavy physical exercise. Majority of reactions are simple, reversible reactions of glycolysis, except three reactions where a special bypass mechanism have been adopted by the cell. The pathways also suffice the much required glucose quota from stored reserves or non-carbohydrate precursors including fat, protein and amino acids. Thank you.